Welcome to the video for 12.6. This is the last section of chapter 12. We will be talking about HIV, the human immunodeficiency virus, which is an RNA virus, but it's what we also refer to as a retrovirus. So we'll be talking about something called reverse transcription. But before we start into talking about HIV, let's do a review question. So review question number 3 states, hypothesize which characteristic of the influenza virus makes the genome particularly susceptible to antigenic shift. Is it A, the genome is comprised of RNA, not DNA? B, the capsid is coated with a host-derived envelope? C, the genome is segmented, or D, the genome is negative sense. So which one of these is going to make it particularly susceptible to antigenic shift? So pause the video, go and flip through chapter 12.5 and see if you can't find the answer. The answer for this is C. Okay, it's that segmented genome that's going to make influenza particularly susceptible to antigenic shift. Now, the fact that it is comprised of RNA, this can help too, but it's going to actually affect more of the mutation rate than its antigenic shift. Okay, so the answer is C. Okay, so chapter 12.6 is about HIV. So we're going to be able to describe the structure of the HIV virion, and we're going to be able to explain the replicative cycle and the epidemiology of HIV when we're done with this section. Okay, so let's start this section with a case history. We have Ralph. He's a 24-year-old who has recently received a promotion at the car dealership where he works. So he's been appointed as head of sales, which comes with a pay increase. Okay, with this pay increase, he's decided that he can support both him and his girlfriend, so he decides to propose. Now, his girlfriend's also working. She works as a bank clerk down the street. So Ralph and his girlfriend have been dating for a little bit, but Ralph had kind of a wild teenage years. He kind of sowed his wild oats everywhere. Now, he's been very upfront with his girlfriend that he engaged in some pretty promiscuous and risky behaviors when he was younger. So before they get married, they've decided to both get tested for STDs just in case they have something. This is how Ralph has learned that he is HIV positive. It's not that the diagnosis came as a surprise based on his past behavior. The surprise was that Ralph has had no symptoms of the disease. So when the diagnosis came, he was a little bit shocked because he thought he'd be sicker than he actually was. Now, one of the first things they do when you are diagnosed with HIV is they look at your T cell count. So a normal human being will have a T cell count about 1,200 cells per millimeter cubed. Well, Ralph's T cell count is considerably lower than this. He's around 500 cells per millimeter cubed. So now that he has this diagnosis and that he's tested positive for HIV, he has a dilemma. How can he move forward with his future plans and getting married if he could possibly infect his future wife? And what about any children they have together? Could he conceive healthy children, or will he pass the virus on to them? So currently, the CDC, the Center of Disease Control and Prevention in the U.S., as soon as you are diagnosed with HIV, the recommendation is to start antiretroviral therapy as soon as possible. Okay, there's a little bit of an argument about this from different physicians and such, but that's the current CDC recommendation. So what Ralph was prescribed is a pill marketed as Truvada. Okay, it's a daily pill that's going to contain 2 drugs that are reverse transcriptase inhibitors. Okay, so they're going to work together to stop the spread of the HIV. Okay, so what's going to happen is it's going to keep the viral levels low. Honestly, this is the advantage of early treatment with antiretrovirals if you're diagnosed with HIV, because things like Truvada are going to stop the decrease in your T cell count. They're going to halt that decline. Now, if you halt the decline of the T cells, we also decrease the transmission rate of and the transmission risk of that virus. Now, there's a lot of physicians that argue with the CDC recommendation. They say that you shouldn't start taking the antivirals as soon as you are diagnosed, but as soon as your T cell count falls below that 500 cells per millimeter cubed. Now, with Ralph, this is one and the same because he's already at that level. One of the reasons for this is that there are some pretty terrible side effects that come with antiretrovirals. But also these antiretrovirals are going to behave as a selective pressure, lot like antibiotics do with bacteria. So the use of these antivirals can select for drug-resistant strains of HIV where Truvada will no longer work on individuals. Now, one of the things that is said in the medical field, it's a standard saying, if HIV is not detected with PCR, then it's not transmitted or not transmittable. Okay, so if we use Truvada and antiretrovirals in order to suppress it, then if we can't detect the virus with PCR, then you're safe to move on with your future plans if you're a Ralph and you're not going to transmit it to your wife or your children. So let's talk about HIV. HIV is a retrovirus, so it has a retrovirus structure. Okay, so it is what is known as a lentivirus. One of the things with retroviruses is they cause infections that progress slowly over many years. So Ralph probably had HIV for a long time, but because it progressed slowly, he really didn't really realize he was sick. Okay, so HIV has a conical icosahedral shape. It is enveloped, meaning it has a cell membrane around it with spike proteins in it. These spike proteins are really integral to its infection cycle. It's going to be what allows it to bind to our T cells. Now, but one of the things that make retroviruses stand out from other RNA viruses is that it has what is called reverse transcriptase. So reverse transcriptase is an enzyme that will take the RNA genome of the HIV virus and it's going to convert it to double-stranded DNA. But the problem with reverse transcriptase is that it's going to have a high error rate. It is as high as it messes up one base for every 10 bases. Okay, we call it a retrovirus, not because it's old. It's not retro in that sense. It's called retro because it is backwards from what normally happens with viruses. So instead of going from DNA to RNA, it goes to RNA to DNA. And the fact that it converts from RNA to DNA, this is why we call it a retrovirus. Okay, but this reverse transcriptase is an integral part of it being a retrovirus. Okay, so let's talk about how it infects T cells. Okay, so T cells have a specific receptor called a CD4 receptor, which is usually near what is called the CCR5 receptor. Now, these are the receptors that the spike proteins on the outside of HIV are going to bind to in order to trick the cell into bringing it in via endocytosis. Now, there's something really interesting because there are some people that have mutations in the CD4-CCR5 proteins, and they are resistant to HIV. They can't catch it because of the mutations in these receptors, or some of them don't even have the receptors with these mutations. Okay, so it would be interesting to continue to study those individuals with those mutations and see if we can apply them and use them to stop HIV transmission as well. Now, so our HIV virion has attached to our cell. Okay, so we're part of CD4 and CCR5. Okay, we're going to fuse with the membrane. And when it does this, it's going to dump all this inner contents out into cytoplasm. Okay, so the core is going to dissolve, and this is going to allow the RNA to be copied and replaced. Okay, so our reverse transcriptase is going to come in and take the RNA and make it into double-stranded DNA. That double-stranded DNA is going to make its way to the nucleus. And one of the reasons why HIV is so deadly is that this double-stranded DNA is going to integrate with the host genome. So if you get HIV, it's a lifelong thing because you can never cut that HIV out of your genome. And it's going to integrate in different places. Okay, so every time the host cell genome is replicated, so is the virus. Okay, so we have it entered. It's going to go through transcription. We're going to make RNA, which are then going to be used to make the envelope proteins. So part of it's going to go to the endoplasmic reticulum, which is then going to go to the Golgi and make these proteins that are going to act as the spike proteins that are going to bind to CD4 and CCR5. Okay, we are also going to make all these other proteins that are going to help rebuild the HIV virus. Okay, the spike proteins are going to be embedded into the genome, just like we saw with the influenza, or not genome, into the membrane, just like the influenza virus. Okay, and then the genome is going to then be packaged, and the new virions are going to bud off here. Okay, a lot of times, instead of budding off, what's going to happen is they're going to go as if they're budding off, and these are going to bind to the CD4, CCR5 of the nearby T cell, and the HIV is actually just going to go from one cell to the next. Okay, so our immune system, well, this is interesting because our T cells are a part of our immune system. So it's actually not HIV killing our T cells, it's our immune system that's killing the T cells because the T cells are infected. This is really, really bad because our T cells are an integral part of our immune system. They're going to be involved in the immune response. We're actually going to talk about the T cells and HIV when we get into the chapters about the immune system. But what's going to happen is the integration of HIV into our genomes is going to prevent our immune cells from being made properly. So we're going to see this decline in our T cell counts. And when we get to a certain level where our T cell counts are so low, this is what's going to lead to AIDS, or that acquired immune deficiency syndrome. Okay, and a lot of individuals who progress to AIDS after being diagnosed with HIV, they're actually not going to die of AIDS. They're actually going to die of a secondary or opportunistic infection, and that's going to be what kills them. This is going to be things like pneumonia. In chapter 11, we talked about toxoplasma, the parasite that we get from cat poop. That can kill individuals with AIDS as well because they don't have the immune system to fight back against that parasite or against that infection that's going to cause pneumonia. So we're going to talk more about this also when we get to the immune system chapters, but let's look at this graphic here. So we have our HIV titer. So this is going to show us the levels of the virus in our blood. Okay, so we have our initial infection where we still have a high T cell count. This is what the blue line indicates. And we're going to have our HIV count. So this is how much of the virus is in our titer. Okay, so after initial infection, we see a rapid increase in the virus, okay, which is going to correspond to a decrease in our T cell count. Okay, but this is what's really interesting about HIV is it can lead to what is called a clinical latency period. So our titer is going to drop off, so we're not going to have as much of the virus, and we're going to still have some, and it's going to just slowly kill off our T cells. Okay, this is what clinical latency is. Now, it's going to progress, so this can happen over a period of years or decades in the case of some individuals. Okay, and then it's going to get to a point where we hit here where our curves cross. Okay, this is around about 350 T cells per microliter. Okay, this is when we start to see problems with our immune system happen when we have been diagnosed with HIV. Okay, so our T cell counts are going to continue to plummet, and our virus levels are going to start to increase because that T cell count is down. Okay, this is the point where we start to see the symptoms like weight loss and fatigue, and we start to see these opportunistic diseases come in. Okay, so we're going to see swollen lymph nodes, we're going to see fever. Our immune system's going to give whatever infection we have a heck of a time, but it's just not going to have enough T cells to actually get rid of that infection. Okay, this is why these opportunistic infections are why the biggest cause of death in those who have HIV that turns into AIDS. Okay, so this is that 350 mark because their immune system is not powerful enough to fight these opportunistic diseases. Okay, they can no longer respond. What's been interesting in the past few years, we've seen that HIV has evolved a bit. It's now making nanotubes in between T cells to travel along. I haven't really heard much about this since then, but this is something that we have we have unfortunately seen. So it makes it even worse because these nanotubes are going to protect it from the immune system. So the immune system's not going to notice it as much. So let's talk about how we treat HIV. Most of ours are HIV therapies, so they're going to be something that's given to you after you have caught HIV. We do have a drug called PrEP, which is going to keep viral levels low. So if you happen to get HIV transmitted to you, your immune system can take care of it before it really integrates into your genome. So it's going to prevent you from getting HIV. If you are an individual that is at high risk of getting HIV. But a lot of our treatments right now are antiretroviral drugs. They're going to target different points in the replication cycle of the retrovirus. So one of the first ones we had was called AZT. We talked about this when we talked about how DNA replicates itself, because AZT uses modified nucleobases. So what it does is it terminates elongation when used. So when they're using it, it would take AZT when they're replicating the viral genome, and it would stop replication. It wouldn't finish elongating the genome of the virus. Okay, so that was one way that we treated it. Unfortunately, HIV has evolved to where this is not an effective treatment anymore. We have all these different ones that are going to do like this one is going to block that CCR5 receptor. We have reverse transcriptase inhibitors, like we talked about in Ralph's treatment. Now, since the start of the HIV epidemic in the 1980s, we have roughly had about 700,000 people die. We have a lot of money going towards research. As I said, we don't have anything but antiretroviral drugs right now. There are no vaccines that are currently approved for it, whether preventative or therapeutic. We do have a few preventative HIV vaccines that are currently in clinical trials, and they're trying to see how well they work. One of them is an mRNA one that came about after the COVID-19 vaccine was developed. But just to put some of this in perspective, we've had 700,000 individuals in the U.S. die since the 1980s. We easily reached that number of deaths from COVID in the first 2 years. So not saying that it's not important that HIV is funded and researched and we find a treatment and a preventative for it, but COVID-19, where it was a brand new thing that caused a pandemic, we saw similar number rates in less time. Okay, so cool fact before we stop talking about retroviruses. When we sequenced the human genome and started DNA sequencing viruses, something really interesting showed up. Our genome is actually riddled. So we have lots of decaying retroviral genomes in our DNA called retroelements. It is predicted that at least half of the sequence of the human genome was originally from viruses and retroelements. It's interesting to think that we're mostly viral, like at least half viral DNA. Okay, the thing is with these retroelements, they're not going to cause disease because the integrated viral genome has mutated enough that it has lost its ability to produce new virions. Now, that doesn't say that there can't be a mutation that comes in that reverses that mutation and allows it the ability to produce virions, but that's a very, very small chance. Okay, but as I said, it's a little bit interesting to think that we have quite a bit of viral genomes in us. Okay, that is it for 12.6. Let's end on a review question. So both HPV and HIV can remain in a latent state for months to years. In this case, the term clinical latency refers to A, the period of time between the initial entry of the virus into the host cell and the appearance of the first symptoms. B, the period of time when the virus is detectable in the host until the disease has run its course. C, a period of time after the initial infection where the virus is not causing symptoms and is virtually undetectable. For D, the period of time between the initial infection and the integration of the virus genome into the host genome. Okay, so pause the video and see if you can't find the answer in this section. Okay, so the answer to this question is C. It's the period of time after the initial infection when the virus is not causing symptoms and is virtually undetectable. Okay, so we know that B is not correct because HIV never runs its course. Once you have it always. It's not the initial entry of the virus into the host cell. Okay, and it's not the period of time between the initial infection and the integration of the virus genome into the host genome, because at this point we're probably having a slight fever. We really don't know. We have something that's not the flu. Okay, so the answer is C. Okay, so that is the end of chapter 12. We will see you in the videos for chapter 13.